Thanks to the organizing committee for having me here. I'm so glad that I'm not really into transplantation, living up to these three great, let's say, parts of medicine that were illustrated to me. Having a background in anesthesiology, I still remember them, but being firmly grounded in academia now, I'm truly happy that I only have to live up to being an expert-centric and not covering all the fields. It was, though, quite an amazing presentation. So we're shifting gears here totally because we're digging into one of the great mysteries of nature, and that is hibernation. So I have a few disclosures, basically dealing with the company I'm working with, Sulfatech, so I got some grants and some fees, and I will discuss some of their compounds. But as I said, I'm an academic, and I'm really true to that adagium. So I want you to imagine that you can preserve organs, not for hours, but for days or even weeks. That will have a profound impact on your practice, but also on your patients and also on your research. So just imagine what it would look like to have organs present a week or even longer. Actually, the solution to that might be under reach, and that is one of the reasons why I study hibernation. Now, basically, this slide tells you a lot about hibernation. I will not go into all the details, but what it shows you are 6 ground squirrels located in our outdoor enclosures, and they are equipped with a temperature sensor in their abdomen, in their belly, throughout the year. So it reads on the x-axis September, October, November, December, etc. So it's basically the year and then the body temperature. The vague, very vague line you see, which is down below, is the soil temperature. So this is Groningen temperature of the soil. It's not as nice climate as Barcelona, I can assure you that. And actually what you see, so somewhere in October, November, the animal starts dropping its temperature. And it does so quite dramatically. It goes from 37, like we are, up till very close to freezing in January, when the soil actually hits around 0 degrees. So they will defend these 0 degrees. But what this picture beautifully shows is that hibernation is not a one state. It is not a state of cooling down. It is the hibernation state, the cooled state, which is called torpor, indicated there, is regularly intersected by very short periods, typically lasting less than 24 hours, of full rewarming of the animal. So, hibernation consists, any form of hibernation consists of torpor periods alternated with arousal periods. And if you're in the right environment, it goes along with huge swings in body temperature, but also huge swings in a lot of other physiological characteristics. And the remarkable thing to me was, when I first saw these type of recordings, these animals do this without whatever sign of organ damage. Now imagine that you would do this to a human or any organ, it will just not survive. Temperature is, though, not the heart of hibernation. The heart of hibernation is that the animal is capable to shut down its metabolism. So this is the European champion hibernation. That's called the edible dormouse, the gliss. And what you see actually in blue is its metabolic rate when it's entering a torpor bout. So it's oxygen consumption, and you see that it suddenly drops in oxygen consumption. And it drops in oxygen consumption by over 90%. Ultimately, they reach 99% of reduction of metabolism. And you also see that the heart rate, purple line, very well goes along with the temperature line. So the heart rate is a very good indicator of metabolic suppression in hibernators. And the body temperature, it just lags behind and is basically dependent on how cold it is outside. And if you're a big animal like a bear, you go down slowly. And if you're a small animal like a mouse or a dwarf bat or something like that, you go down very rapidly. But temperature is not really this relevant to hibernation. It is the metabolic suppression which is relevant to hibernation and which may be, in my view, be very relevant for your field of work. So you can also do this in the hamster. That's our workhorse. And you can easily track what's going on in the hamster just by placing them in a passive infrared monitoring. So basically, that's the thing that makes your garden lights lit up when you drive past or when you walk past. And what you see here is what's called an ectogram. And every line is 24 hours. And every black bib is when the animal moves. So we first had the animal in autumn conditions, so it was still under 20 degrees, and some light, 10 hours of light a day, and then we switch it to winter conditions. Immediately you see that the animal becomes a lot more active to keep itself warm, because it's now in 4 degrees and in total darkness. So then we gradually decrease the availability of food, and at a certain point in time, in the right side of the slide, you see that there are days without movement. And these are actually these torpor bouts. So you can very easily follow this. And for all of you who have kids and a hamster, I recommend that you have a passive movement device installed. Put the hamster in the garage. There it's dark. When it's below 15 degrees, it will show these patterns. So it's very easy to sort of do in a hamster. And when you do this, another remarkable feature of hibernation comes to life, and that's here. That's the arousal part. So basically what you see here is a hibernator caught on thermal camera. So the lower one, it reads 6 degrees, and the higher one, it reads 28 degrees. That's normal skin temperature of a hamster. So you see it here in its burrow, and all the time I'm speaking, every second is a minute. And basically you see its head down, so the head is towards us, and what you now already start noticing is that the head shows a bit of a yellow glance to it. And that will absolutely amplify over the next seconds, because the head of the animal is what warms up first. He has special patches of brown fat located around the major arteries to its head, and it uses it to rewarm the brain at enormous speed. So basically here you see the brain is more or less rewarmed, and still the hindquarters are very, very, very cold. So the animal goes on, continuing like this. You see some eyes peeking out. They are really, really hot. And at a certain moment in time, the front is warmed enough for the animal to turn around, and it makes use of everything it's heated there in the burrow to rewarm its hindquarters, and then it's good to go. So basically, this whole process, I'm talking now for 1.5 minutes, so this takes 1.5 hours, of which basically the first 40 minutes you don't see anything. And this is an astonishing rate of reperfusion or rewarming in an animal. If you do this to organs, that is really, and for sure to whole persons, that is unmet. You cannot do this in a medical setting. And these animals, remember, they do it 20, 30 times per winter. So there's a lot to learn from hibernating animals. And you may think, yeah, okay, hibernating animals, these are the freaks of nature, but this is the phylogenetic tree of the mammals. And wherever you look in the phylogenetic trees of mammals, there are hibernators. For sure, up to your left, the rodents, but also the one below. These are primates. That's us, right? So there are by now 4 primates known who hibernate, who are capable of hibernation. So probably, and most likely, we still have this program also in us. Now, dependent on your body size, how much energy you have available, how much you need, hibernation can take different forms. It has different names, but it all boils down to the same. So some primates just go with the temperature that's on the left, this lemur. Some go into a daily hibernation. That is a mouse. And for everyone here in the audience who does mouse experiments, I don't think you very often recognize that mice are capable of daily hibernation. And in fact, part of your results may be explained by this. And of course, we all know the bear, and the bear stays around 32 degrees in body temperature. But if you look at these animals, they all have periods of vastly suppressed metabolism, here indicated by the red dots. So they all deploy some form of torpor alternated with arousals. So let me summarize hibernation. It is a suppression of metabolism, and in essence, its mechanism is unknown. So there's a Nobel Prize to gain there. It features large and very rapid physiological changes. And here in the table is just a hamster, which has a body temperature normally of 36, drops down to 8 or lower even. But the amazing thing is in the heart rate, ventilation rate, and blood pressure. In summer, they have a heart rate of 325, it drops down to 11. They have a ventilation rate of 60, it just drops down to 2. If you would see a hibernating hamster, you could not tell that it is breathing. You really need an expert eye for this. The blood pressure also drops enormously down. But during these arousals, it swings back, and it even has a sort of a compensation, and that's because the animal is in very cold temperature. So basically, it swings from a almost standstill into running a marathon and back and forth 30 times per winter without any organ damage. So some years ago, we asked ourselves the question, these cells of hibernators, are they specially equipped to deal with this cold? And actually, yes, they are. And here is an experiment which shows this. And I'm not going to cover all the details of the experiment, but I'm just going to give you a flavor of it. So basically, what these hamsters can do is they maintain hamster cells. These are isolated cells, not a whole animal. These hamster cells, if you cool them down, for instance, on the left, 16 hours at 4 degrees, they maintain ATP synthesis. And in the max at production of reactive oxygen species, you see when cooling down these cells at 2 hours, and you also see that the hamsters in black do not increase in ROS production. So they deploy some sort of defense mechanism which make them be able to cope better with cooling and rewarming compared to us, to our cells. I mean, that's the gray ones, we start immediately to lose the capability of synthesizing ATP, or we start producing ROS. Or I should rather say, and start producing ROS. So many years ago, we took this as a very simple fact, and then we said, okay, what can we do with this? So we explored hamster cells on your far left, and went and have them in a cooling rewarming paradigm. So 24 hours cooling at 4 degrees and 3 hours rewarming at 37. Basically, this is an experiment you can just do at home, because the 4 degrees sounds very fancy, but it was basically store them in the fridge. So you can do that at home. And if you do that with hamster cells, you see that all of the hamster cells survive. That's the black bar as opposed to the gray one. But if you do that with human cells or rat cells, typically half of them die. So you see that all of these cell lines indicated and cells indicated decrease viability by about 50%. Then we did a very intriguing experiment. We cooled and rewarmed hamster cells, and then we got the medium of these cells. And we transferred this conditioned medium to rat cells and human cells, which we then again forced into a cooling and rewarming cycle. And what then happens is that the conditioned cells increase viability. So there's something in the medium of the cultured hamster cells, if you cool and rewarm them, that is protecting all types of other cells. It took us an enormous amount of time to find out what this was, and this is the mechanism. You can read about it in this publication, but the mechanism is as follows. As soon as you start to cool the cells of hibernators, they excrete serotonin. And they do not all use serotonin. There is also a number of other compounds they use, but this particular cell line uses serotonin. It excretes it. It's been taken up again, so this is the reuptake transporter of serotonin, and then it's delivered to an enzyme which is called CBS, cystathionine beta synthase, and that is an enzyme capable of synthesizing H2S. And we think that this is really a mechanism which helps these animals to survive this. And this is, for instance, corroborated by, on the top left, hamsters having a high H2S synthesis, on the top right, being able to induce H2S synthesis with serotonin in red cells, and on the left, if you knock out the CBS, you don't see any effect of the serotonin anymore. So we think this is an important mechanism. Now, knowing this, you could easily use it, right? Well, the problem is that these compounds that do these tricks, like the serotonin, but also dopamine works along the same lines, and even histamine, adrenaline, all these bioamines have a huge amount of receptors in organs and in our bodies. So you suffer a lot from side effects. So basically what we did is we knew these compounds, and what we did next was, and I'm not sure why there's all these strange marks around it. So basically we took these compounds and we screened the library of backbones, and we ended up with 2 families with quite a lot of lead compounds. And all within these lead compounds, we characterized a whole bunch who are capable of increasing viability when cooled. So basically what you see here is those response curves of cells cooled, and then you see that their viability drops down to 0. And then if you start to treat them with increasing amounts of these compounds, you see that their viability goes up. The absolute level is not the message here. That depends on how many cells are present. But the fact that they reach a plateau is indicative that they are actually preserving cells. So we call these compounds 6-hydroxychromanols. This is their chemical structure. So it's basically a simple molecule, easy to use. So what these compounds do is, first, they preserve the membrane potential of mitochondria in the cold. And by doing so, they lower the reactive oxygen species. And they do so by very specifically working on 2 complexes of the mitochondria, complex 1 and complex 4. And basically what they do is, if you frustrate these complexes in the mitochondria by giving inhibitors, they are able to again reactivate these complexes, resulting in increased ATP and lowered ROS production. In the end, it also works in a rat. Here we took it to a rat, cooled the rat for 3 hours to 15 degrees, harvested the organ, and the only thing I'm going to share with you is a view at their kidneys. So if you look at a normal uncooled rat, everything looks okay. But if you look at a rat who's just only cooled, you see, for instance, in the middle upper section that the glomerulus is gone, that the tubuli are basically gone. And the compound sort of rescues it all. The same goes for tubular damage and for the influx of all kinds of immune cells. So I want to summarize by first implanting in your brains that hibernation has very cool features. These hamster cells are protected, and they are protected by a mechanism which maintains their mitochondrial function in the cold. And we think that this serotonin-dependent activation of this pathway producing H2S offers a great deal. So the hibernation is a biomimicry platform for drug development, and we developed this 6-hydroxychromanols. They support the mitochondrial function and they preclude cooling rewarming, and I'm looking forward to see what that would mean to the transplantation field. So I want to acknowledge a lot of people on which we work with on hibernation. They're basically on this slide, and with the message that hibernation has much more in store than only cold. It also features immune suppression. It also features anti-clotting. I sort of invite me back to this wonderful stage in a couple of years. Thank you very much.
Robert, thank you so very much for sharing in such an articulate way your experience and expertise for this fascinating topic. Our iPads are heating up with questions. Please keep them coming. But the prevailing one for your presentation is very clear. If the animals have it, why we don't have it? Yeah. And if we had it and we lost it, can we get it back?
I think that we might have lost some key switches. I think that we are capable of hibernation up to a certain extent. But what we did in evolution is sort of adapt ourselves in coping with a hostile environment in a different way. Rather than going into hibernation, we start building houses and burning fuel, basically this. But I think that I already showed that parts of this is still present in us, and we may actually tap into this to sort of use it for questions like in the transplantation field.
Thank you very much. Ladies and gentlemen, Professor Robert Henning.